Okay, hello. Good morning, everyone. My name is Sam Miller, and today I'm pleased to be able to give you guys a little bit of a genetics background onto the into the insights into NARS1 and the genetics, the symptoms, and the family perspectives of this disease that you guys are all, to some varying level familiar with. Today, I'm doing this in collaboration with the Rory Bell Foundation, who has been a great resource to help me kind of gain a insight into this rare disease community and the University of Pennsylvania, for which I'm currently a master's student there. So who am I? Like I said, my name is Sam Miller. I graduated from the University of Delaware roughly 4 years ago with my bachelor's in biological sciences. Since then, I spent 2 years researching immunotherapy at the University of Pennsylvania, and this really allowed me to get a introduction into the importance of scientific research and furthered my passion for becoming a genetic counselor. And so currently I am just finishing up my first year as a master's student at UPenn studying genetic counseling. I'm currently like potentially interested in working in the field of pediatric genetics, and that is kind of really what brought me to this rare disease foundation. I think that there's a great, it's a great like resource for patients who and their families who are impacted by NARS1. So I'm, pleased to give this presentation to you guys today. Thank you. So just getting into it, I wanted to first talk about kind of an overview for our genes and our genetics, and how these play into the role of who we are as people, and how this can go forth and create problematic diseases when there's like issues in our genes. So just at the very base level, all of us have this molecule inside of our cells called DNA, and DNA acts as instructions for our cells to create all of the products that are necessary for them to properly function. And so our cells have roughly 20,000 different genes. And so all of those genes are packaged into distinct instruction manuals, if you will. And furthermore, all 20,000 of those have to fit into each tiny little cell. So those are all packaged into our chromosomes, of which we have 23 pairs, 46 in total. And again, like I said, those all are able to be read like an instruction manual, and then the cell is able to create functional products with that, and allow the cell to function properly. And then together, all of our cells allow our bodies to move and operate how we would from day to day. So looking at kind of like where we do get our DNA, and kind of the genes that our body uses to function. As you might all know, we get half of our genes, or the chromosomes that I said that I talked about earlier. Half of those come from our mom, and half of those come from our dad. So here you'll see on the left here the ovum, which is the egg cells that come from your mother, have 23 chromosomes, and your father also has one set of 23 chromosomes. And so you'll see here this first big one here from dad matches up with the first big one from mom, and those work together to give you two different. So all of the genes on that chromosome, you have one copy from mom, one copy from dad. And when the egg and the sperm combine, you get what's called a zygote, and that's kind of the earliest cell that forms when we are developing as a baby. So going forth, I wanted to talk to you a little bit about inheritance and how the traits that we have are showed like once we start developing. So traits are kind of distinct features. So you might know a lot of them as like hair color or eye color, how tall you are, etc. And these traits are shown in different manners. So one trait might be um, quote unquote like more powerful than a different trait, so it's more likely to be presented. And so these are what are known as autosomal dominant traits. I'll just refer to them as a dominant trait from here on out. And some traits you have are known as recessive, and so uh, those need both copies of the gene to be in order to be presented. So in an autosomal dominant trait, let's just say, for instance, brown eyes. People with brown eyes only need one single copy of like a brown eye gene for that trait to show. So in this case here, you have somebody who has two, let's say, blue eye genes who has blue eyes, and the other affected parent over here has brown eyes because they have one brown and one blue, but the brown is preferentially expressed. And so when they have children, you'll have two children who have blue eyes, and you'll have two children who have brown eyes. When thinking about a recessive trait, though, you have two parents here who both have brown eyes, but you'll notice that each only have one brown and one blue trait. So when they go to have children, the possibilities for those children are they would have, let's say, like if this is blue eyes, you would have one child that has blue eyes, um, two children who have brown eyes. Sorry, let me reorient myself here. So when talking about autosomal recessive traits, I think maybe it's a little bit more pragmatic to talk about NARS1 itself. So NARS1, as you guys might know, is a autosomal recessive inherited disease. Um, so in this case, you need two copies of like a non-working or like a damaged NARS1 gene in order to have a NARS1 phenotype or symptoms. So in this case, you'll have, let's say these are both the NARS1 genes. So here you have one working and one not working, and the other parent you again have one working, one non-working. And so when they go to have children, you'll have one child with both working copies. They're totally fine. Two children, or half of the children rather, would be just like the parents. They'd have one working copy, one non-working copy, but otherwise they wouldn't be symptomatic. And then in one of four children, you would have an affected child with NARS1 because both copies of NARS1 would not be functional. Another way in which patients can present with symptoms of NARS1 is they have what are called de novo variants. So here you have two parents who are unaffected. They both have, properly functioning NARS1 genes that are highlighted by these blue bands and then also green bands. And so you see that those parents both pass along one chromosome, so that this parent passes along their blue chromosome, this parent passes along their green chromosome. You can see the green NARS1 gene here is totally fine, but you'll see that in the blue chromosome, there was a mutation that caused for that copy to not function properly. And so this specific mutation somehow was able to overpower the other one, and this child would present with NARS1. Going forth, just taking a look at the NARS1 gene itself. So you'll see here, if we can imagine a gene kind of like a long strand of letters being read from kind of this left side here till the end at the right. There are 548 kind of little blocks of information in that. And so those blocks each tell the cells or kind of signify one amino acid or one protein to fit in there. So as you can see, there's 548 different amino acids that get added to this protein to make it one big functional protein. And you'll see here all of these, P dot, let's say NET69, ASNFS, like all of these are different positions at which the gene has variants that cause NARS1 to occur. And so you'll see a lot of different things, like you'll have about one third of these in people that we see are the recessive. So both parents pass down a non-working copy, and that ends up being the situation where their child has a non-working has symptoms of NARS1. There can be another instance where both parents pass down um, a mutation or a variant that aren't the same, but because both of those don't work in a different way, the child will still have symptoms of NARS1. And so those are called compound heterozygotes. So compound just being two different, but they work together to create kind of the disease symptoms that we see. And then finally, about one third of these are the de novo cases. So this is where neither parent has, any damage to their NARS1 genes, but when the child was developing, there was some sort of random event that happened that caused them to get NARS1. So typically, when looking at genetic testing for NARS1, these children often are presenting with things such as seizures or speech delay. Some of them might have like global developmental delay. And a lot of these symptoms tend to overlap with other genetic conditions. And so it's possible that because these symptoms are seen in a lot of different conditions, that multiple rounds of genetic testing might be required to find a NARS1 variant, especially because NARS1 is relatively new and people aren't necessarily super familiar with its role in causing any sort of disease. So testing might include karyotyping, and so that includes just looking at someone's chromosomes kind of on a large scale. And so they'll see if there's anything there that's like a big chunk is missing, or there's like an extra material there that's not supposed to be there. So that's typically like your first line of testing. If they don't see anything there, they might move to like a panel of genes. So this might include something like like an epilepsy panel, um, or something like that, that is looking at a set of genes that are recognized to cause seizures. And so if NARS1 is like a really new gene, you can imagine that it might not be on that type of panel. And so finally, what probably happens for a lot of patients who have NARS1, exome sequencing is sent for them. And so what this does is it looks at all of the genes inside somebody that code for a protein, and it looks at kind of matching up the symptoms that they have with any sort of known gene that can cause that type of symptom, and it therefore is able to pick up a lot more than karyotyping or a panel is able to pick up. And then just quickly, I wanted to also go over the types of variants that people will see. So I think most commonly people either get a pathogenic variant. So this is where the variant is known to be associated with a specific disease or a condition. And it often has a lot of functional data that supports this, and the geneticist is often very, what's the word? The geneticist is also feeling very, confident that this is, the reason for somebody's symptoms. On the opposite end, they get what is called a benign variant, and so this is what we consider normal variation in the population. So as you all know, we all have different variants in our DNA, and that's why we all look different. But just because we all look different doesn't mean that our bodies are not functioning properly. So these benign variants are totally normal. They don't cause the gene to act in a different way, and they're totally fine. Somewhere in the middle of these two extremes, we have what are called variant of uncertain significance, or VUS. So these are basically just there's not enough uh, either not enough data, or there's either conflicting evidence to support whether or not it's these variants cause any sort of type of disease. And so in a gene like NARS1, where relatively little is known, it's kind of common to see a VUS. And I think what's really important to know about this is that um, VUSs, as we learn more and more about our genes, they can be reclassified at a later date as more data comes in. So it's important to speak with your geneticist or your genetic counselor that you have once every, I'd say, like 2 to 3 years to just see if there's any updates if you do have a VUS. And you can also enroll your child's VUS at the Brain Gene Registry, and they will automatically send updates on any changes that occur. And that's also whether or not it gets upgraded to pathogenic or it gets downgraded to benign. Again, that's a great resource. They look into like doing a lot of research with kind of different symptomatic like brain diseases. So I know this is kind of maybe disorienting for a lot of people. So I just wanted to give a little bit of an overview of the NARS1 gene and how it works and why you see a disease um, when this protein or the gene is damaged. So NARS1 is a gene that tells our bodies to make a protein, which is called a tRNA synthetase. And so I have up here that is kind of marked by a little like loading dock construction worker. So the tRNA synthetase's worker, their job is to load the proper materials onto the tRNA shipment truck. The materials that each tRNA synthetase adds is a specific amino acid. So in the case of NARS1, it adds the amino acid asparagine. And so these materials are loaded onto their tRNA. The tRNA is responsible for delivering that specific package or that specific amino acid to the protein construction site, which is called the ribosome. And so the ribosome, like I just said, it's adding each separate amino acid that the tRNA brings, and altogether that creates like a larger protein. And so, like I said, the growing protein chain, it's very necessary that they have all the proper materials to build the growing protein, just like it's necessary that we have all the materials to build a building, such as brick, mortar, steel beams, and everything. And so having all these materials allows for our bodies to function, grow, and develop effectively. In people with NARS1, the important role of this tRNA synthetase construction worker, it's not working either at all, or it's damaged to a point where it's not working as efficiently. And so the impact of this is that the ribosome construction site either isn't getting materials at all for that one amino acid, or maybe it's getting a reduced amount of that amino acid. And so the construction site often has to either just fill that in with different amino acids, so that has that can cause problems by adding like a brick where maybe steel was supposed to go, so that can cause an issue. Or they could just not put anything there, and any protein that's being any protein that uses asparagine might not get made at all. So you can think there's thousands of different proteins. If 500 of those use asparagine, then you have 500 or half of your proteins that are affected by this. Just thinking of some of the uh, symptoms that come along with this. I kind of have a like multiple broad areas that are impacted. So you have issues with communication, neurological or like cognition, behavioral, uh, or emotional issues. Some of those are like kids might be hyperactive, they might be impulsive. Um, I've like heard that they're very social and friendly. Um, there could be some musculoskeletal issues, so there might be I think like I've heard of like joint laxity, so their joints might move, further than what we would expect them to. Um, as for gastrointestinal issues, that's like issues with like feeding or constipation. They might have issues with eating at all. Um, and so because of that, a lot of these kids might have growth issues because they're not getting all the necessary nutrients that they need to grow. They might have sleep issues. They might have issues moving around or walking. And then, some other like in some cases they might have there's like visual and or auditory issues as well. Here I just wanted to kind of give the distinct signs and symptoms of these for both the kids who inherit this in a recessive way, whether that's two of the same two of the same mutation that come from one of each, come from both parents, or if they're getting one variant from one parent and a different variant from another parent, but together those are causing this disease. And so that's again the compound heterozygous kids. And then comparing both of those recessive variants to the de novo variants. So here, like I've kind of set up in this chart, you can see both have global developmental delay. Um, Typically, in all of these cases, in all of these different symptom areas, we have a more severe form in the autosomal recessive than we do in the de novo. So just going down, we're looking at seizures. Um, with the recessive kids, you'll see a greater range and severity. So they have tonic-clonic seizures, febrile seizures, focal seizures, generalized seizures, etc. In the de novo, you typically only will have like febrile seizures, and those are typically treated, more effectively with medication than they are for the recessive kids. Um, in speech and language, you'll typically have severe delay with the recessive people, and then more moderate delay in the de novo ones. Those individuals might use a few words by the time they're toddlers or like young children. The GI issues are pretty much kind of going along that same trajectory. They both experience feeding issues, constipation, pain when eating. Um, but again, the recessive people will often have more severely impacted by this, and they might have poor growth because of this. Um, and then lastly, I just wanted to kind of talk about hypotonia, which is kind of like poor tone, or like they might seem weak, or ataxia, which is like loss of coordination. And so because of this, the recessive uh, people with the recessive variants might fail to walk and have like a severe delay with moving around in their environment, whereas the de novo kids uh, might experience some like more mild delay, but eventually, maybe catch up or at one point, walk with some aid. And then I think it was also important to me as I went through this internship um, to kind of also reflect on some of the information that I found out from talking to and interviewing some of the patient families. And I think it's important to give some caregiver testimonies that I kind of picked up along the way. So I think something that I thought was really impactful was the like varying use of different therapies. Um, that people would use to help their children thrive. And so I think it's really important to touch on here how, some people really just did what I think it's important to kind of leave everyone here with the notion that like you have to do what's important for what works for your family. So I think like some people people would do like uh, would use like a combination of occupational therapy, physical therapy. They would do speech therapy. It's like all at the same time, whereas others might choose to like work on OT this month and then do PT the following month, or take short breaks in between like different therapies. And so I think it really just depends on what like works for your individual child. And I think that, you know, certainly, a lot of this has an impact uh on like the well-being of parents as well. So I think like you also have to take care of yourself as like as a caregiver as well. Some other things that I, noticed were, people had a poor experience with their diagnosis. They had a lot of different specialists who, couldn't give them the answers for their child's disease. And so I just wanted to like recognize that there's a lot of, stress that goes into your child is, um, maybe getting worse as time progresses, and nobody has answers to give you to help them. So I think that was a common like theme that went along with this. Something that I thought was unfortunate was that genetic counseling seemed wildly unhelpful, and that's like again their genetic counselor didn't know anything about the disease course, or any like gave any resources that would be helpful. So that was really unfortunate, especially given that I'm looking to go into this field. Um, and then I think something kind of hopeful that parents gave were that children with NARS1 uh, will, I think, push the boundaries of what they expected them to do. And so it seems that they're all eager to persevere in their own maybe unique way. And I think some of them have shown that they're, more independent than I think their parents would have thought. And so I think that's, really powerful. Um, Going into the next section, I just wanted to talk about some support and therapy. So, of course, you guys have heard of the Rory Bell Foundation. They're a great group. I think it's a really important resource that everyone who has a child with NARS1 use because it creates a sense of community. Some of the other groups that that are out there, like Rare X, who looks into kind of compiling a bunch of different rare diseases, and they use the collective power of all these rare diseases to create kind of an infrastructure to get more treatments for these people with rare diseases, and do like more research with that. Um, I'll talk about the National Brain Registry I talked about earlier. They were, kind of very similar to Rare X, but they are specifically looking at stuff that has kind of like a neurodevelopmental symptoms that come along with that. Niho, I think, was really cool. That was actually a dad who has a child with a rare disease, and he basically created this like app that allows you to compile all of the information on your child, and it can be anything from medical information that's relevant to their doctor visits, all the way to like they don't like bologna sandwiches, and so like if you have a caregiver, maybe they experience pain when eating like sandwiches of any kind. And so you would let their caregiver know that like, hey, they can look at this app and they will know that like, hey, they don't eat sandwiches because it's not pleasant for them. So I think that was really cool. Um, just looking at some of the therapies that are out there to help these children. I think some of the traditional therapies, like I spoke of earlier, are physical therapy, occupational therapy, which kind of allows them to navigate their daily life a little bit better, and speech therapy, which will help them to communicate with with their caregivers, and maybe help them to experience less frustration with getting the things they want. And then some other therapies that I've heard about, that just are able to maybe improve the quality of life for these children are, something that I thought was interesting with hippotherapy. So I guess that's horse therapy. I'm not exactly sure whether they take time riding horses or they're just like around these horses, but I've heard that they have a really good positive experience with and or around these horses. aquatherapy. So they could like the child enjoys swimming or just being in the water is very like calming and relaxing to them. There's also like noted use of music therapy, vision therapy, especially for children who have um, like symptoms that impact their eyes or their vision, and then also action behavior analysis, which is typically used for children who have a diagnosis of autism. So some people will get that like secondary diagnosis with this and are able to use that really good therapy option as well. And then lastly, just to close this out, I wanted to kind of talk about genetic counseling. Being a student, I thought this was really important, and I think that despite the maybe negative experiences thus far of people with NARS1, I think the National Society of Genetic Counselors is a really good, kind of structure and goal of, helping people process and understand the medical and the psychological and the familial implications of, um, having a genetic disease and how this impacts not only the individual, but also, the wider family, social setting, and how, we can help them work through that. Um, and I think it's just like a really great time it's a, I think for me, I think like the most rewarding part about this is that we as genetic counselors get to educate people about inheritance, testing, how they can best manage their symptoms or the symptoms of their child, um, how to prevent, these if you don't want to have future pregnancies that are impacted by this, how we can connect them to resources, and maybe even get, research or promote research that can help them create like better informed choices and adapt to like the different risks of this condition. And then with that, I just wanted to say thank you to all the people who work every day to help these patient families and who are doing research into helping the people with NARS1 and the caregivers of this disease. So it was a really great time working with you guys and getting to interview everyone that I was able to. And with that, I just wanted to say thank you.